Hello, this is Professor Beda. Today we will be doing chapter 3, which is the chemical building blocks of life. The bodies of organisms contain thousands of different kinds of molecules. These enormous biological macromolecules are built up from smaller compounds with simpler chemical structures. Each of these basic compounds consists of a framework of carbon atoms bonded to other carbon atoms of oxygen, nitrogen, or sulfur, phosphorus, or hydrogen. A chemical compound containing the element carbon is called an organic or an organ molecules. Because carbon atoms can form up to 4 covalent bonds, molecule containing carbon can form straight chains, branches, or even rings, balls, and coils shapes. Hydrocarbons, those are molecules consisting of only of carbon and hydrogen. Carbon and hydrogen atoms both have very similar electronegativities. Electrons in CC or in carbon-carbon and carbon-hydrogen bonds are therefore evenly distributed with no significant differences in charges over the molecule surface, for those are non-polar molecules. If you remember that, we explained this. Non-polar molecules, they don't have different charges on the same molecule. And of course, they have different or they have functional group added to chemical properties. Most biological molecules produced by cells, the hydrocarbons, also contain other atoms. Because these other atoms have different electronegativities, molecules containing them exhibit regions of partial positive or negative charge that are polar. So these attached atoms group called functional groups. Functional groups, we have different functional groups or those molecular group. They attach to the hydrocarbons molecules that confers chemical properties or reactivities. They are responsible for the chemical reaction for hydrocarbon. We have hydroxyl group, carbonyl, carboxyl, amino phosphate, and methyl. Isomers are compounds with the same molecular formula but different structures and properties. There are two types of isomers, structural isomers and stereoisomers. So structural isomers which differ in the actual carbon skeleton, and stereoisomers which differ in the spatial arrangement of groups attached to a carbon skeleton. When the 4 groups attached to a carbon atom are different, the resulting molecules is said to be chiral. So chiral molecules can have mirror images to each other. We call the enantiomer. Enantiomers are isomers that are mirror image of each other and that differ in shape due to the presence of an asymmetric carbon. Then the 4 groups can arranged in space around the symmetric carbon in a symmetric carbon in 2 different ways that are mirror images. Enantiomers are, in a way, left-handed and right-handed version of the molecule, just as your right hand won't fit a left-handed glove. A right-handed molecule won't fit into the same space as the left-handed version. So usually only one isomer is biologically active because only that form can bind to specific molecule in an organism. Biological macromolecules are commonly grouped into four general classes: carbohydrates, protein, nucleic acids, and lipids. The first three of these are all polymers constructed by joining together many small similar chemical subunits called monomers. A long polymer of linked monomers form a molecule analogous to a long train consisting of many railroad cars linked together. Different uh, polymers are determined by their constituent monomers. Complex carbohydrates such as starch are polymers composed of simple ring-shaped sugar, and nucleic acid, DNA, and RNA are polymers of nucleotides, and proteins are polymers of amino acids. The class of macromolecules that don't fit this simple monomer-polymer framework are the lipids. This diverse group of lipids of the lipid molecule, uh, those molecules shares the chemical features of being hydrophobic and include fats, oils, and steroids, each with different function. The formation of carbohydrates, proteins, and nucleic acid are all built in the same way, assembled via chemical reaction, some dehydration reaction. So the basic chemistry of their synthesis is similar. To form a covalent bond between two monomers, an uh, hydroxide group is removed from one monomer, and a hydrogen atom is removed from the other. Uh, and this reaction is condensation reaction or dehydration reaction. The hydrolysis reaction, cells disassemble macromolecules into their constituent subunits through reactions that are the reverse of dehydration reaction. A molecule of water is added instead of removed. In this reaction, called hydrolysis reaction, a hydrogen atom is attached to one subunit and hydroxyl group to the other, breaking a specific covalent bond in the macromolecule. Carbohydrate is a group of molecules that contain carbon, hydrogen, and oxygen in the molar ratio 1 to 2 to 1. The empirical formula is C2O by N, where N is the number of carbon atoms. Because they contain many carbon-hydrogen bonds, uh, which release energy when oxidation occurs, carbohydrates are well suited for energy storage. Sugar are among the most important energy storage molecule, and they exist in several different forms. The simplest of the carbohydrate sugars are the monosaccharides, and this is derived from Greek word mono mean single, and saccharum or saccharides mean sugar. Simple sugars contain as few as 3 carbon atoms, but those that play the central role in energy storage have 6 carbon sugar. The empirical formula of 6 carbon sugar is C6H12O6. 6 carbon sugars can exist as straight chain, but dissolved in water. When they dissolve in water, they always form rings. The most important of the 6 carbon monosaccharides for energy storage is glucose. Glucose has 7 energy storing carbon hydrogen bonds. So monosaccharide or simple sugar can contain as few as 3 carbon atoms and are often used as the building blocks to form larger molecules. The 5-carbon sugar, ribose and deoxyribose are components of nucleic. So glucose is a linear 6-carbon molecules that forms a 6-membered ring in a solution. They form a ring in a solution when they dissolve in solution. Ring closure occurs such that two forms can result. So alpha glucose and beta glucose, and the structure they differ only in the position of hydroxyl group bound to the carbon number 1. They differ in the special arrangement of the hydroxyl group. So these differences often account for substantial functional differences between the isomers. So these two different isomers. Your taste buds can discern them. Fructose tastes much sweeter than glucose, despite the fact that both sugars have identical chemical composition. Enzymes that act on different sugars can distinguish both the structural isomers and stereoisomers of this basic 6-carbon skeleton. So glucose is not the only sugar with the formula C6H12O6. Both structural isomers and stereoisomers of this simple 6-carbon skeleton exist in nature. Fructose is the structural isomer that differ in the position of carbonyl group, okay, as you see here, and galactose, stereoisomer that differ in the position of hydroxyl group and hydrogen group relative to the ring attached to the carbon number 4. Both animals and plants transport sugars within their bodies. In humans, the glucose that circulates in the blood does so as a simple monosaccharide. In plants and many other animals, however, glucose is converted into transport form before it is removed or it is moved from place to place within the body. In such a form, it is less readily metabolized during transport. So transport forms of sugars are commonly made by linking two monosaccharides together to form disaccharides, such as sucrose, lactose, and maltose. Polysaccharides are longer sugar polymers made up of monosaccharides that have been joined through dehydration reaction. Organisms store the metabolic energy contained monosaccharides by first converting them into disaccharides. These are then linked together into insoluble storage polysaccharides called starches. In plants, we call those storage polysaccharides or sugar as starch. The molecule comparable to starch in animal is glycogen. Polysaccharides also found as structural compounds that support cells or organisms. In plants, the polysaccharides found in the form of cellulose, and in animals, in arthropods and fungi, use chitin. Those are forms, chitins and cellulose. Those are forms of polysaccharides, carbohydrates. Stars differ mainly in how the long chain polymers branch and unbranch chain. So we have two types of starch. One simple called amylose, and the other is branched and complex we called amylopectin. Glycogen is found in animal cells and is highly branched and insoluble, of course, forming glycogen granules. So starch chains consists of alpha glucose subunits, and cellulose chains consist of beta glucose subunits. Cellulose is unbranched and forms long fibers. Cellulose fibers can be very strong and are quite resistant to metabolic breakdown, which is one reason wood is such a good building material. And we have also chitin. Chitin is structural material found in arthropods and in many fungi. Proteins are the most diverse group of biological macromolecules, both chemically and functionally. We can group these functions into the following 7 categories: enzyme, catalysis. Enzymes are biological catalysts that facilitate specific chemical reaction. They speed up the chemical reaction. In defense, proteins, we have an example: recognizing foreign microbes and cancer cells. White blood cells destroy foreign cells and other by making antibodies, proteins. Those are proteins and antibodies that attach to invading cells. In transport, we have an example of globular proteins transport small molecules. We have red blood cells, for example, contain the transport proteins or globular protein hemoglobin that transport oxygen in the blood. In support, proteins fibers play many important structural roles. In human, these fibers include keratin, the fibrin in blood clot, collagen forms the matrix of skin, ligaments, tendons, cartilage, and bones. Proteins function in motion. We have an example in our muscle cells. Muscles contract through the sliding motion of 2 kinds of proteins filaments. Those are actin and myosin. In regulation, proteins play, or the small proteins called hormones, serve as intracellular messengers in animals. In storage, proteins play important role in storage. For example, calcium and iron are stored in the body by binding as ions to storage proteins found in cells. Proteins are polymers, linear polymers of amino acids. Proteins composed of 1 or more long unbranched chains, and each chain is a polypeptide. Amino acids are monomers, or the building blocks of a protein. Amino acid structure consists of central carbon atom, amino group, carboxyl group, single hydrogen, and variable R group. R groups determine the chemistry of amino acids, and it could be nonpolar, like leucine, threonine, glutamic acid, phenylalanine, proline, and cysteine. Those are types. We have 20 different types of amino acids in our body. So the R group, it's maybe nonpolar, polar uncharged, charged, aromatic, unit. This figure from your textbook shows the different 20 amino acids found in nature, and these 20 different types form different types of proteins according to their sequence within the proteins or the polypeptides. So the amino acids or the amino groups on one amino acid, the amino group or one amino acid can undergo a dehydration reaction with the carboxyl group on other amino acids to form covalent bonds. The covalent bond that link two amino acids is called peptide bond. Okay, peptide bond is formed when the amino acid or the amino end of one amino acid is joined to carboxyl end of the other amino acid. Note that the resulting dipeptide that means two amino acids linked together via peptide bond, has an amino group end and carboxyl end. So it still has these two ends. The final folded shape of globular protein is called its tertiary structure. So, four levels of protein structure. The primary structure of a protein is its amino acid sequence. A secondary structure, instruction of groups in the peptide backbone. If the peptide were coiled into a spiral, that were called an alpha helix. Occur between regions of peptide aligned next to each other to form a planar structure called beta sheet. So we have two arrangements of or two forms to the secondary structure, alpha helix and beta sheet. The final folded shape of a globular protein is called a tertiary structure. The tertiary structure of a protein is stabilized by a number of forces, including hydrogen bonding between R group of different amino acids, electrostatic attraction between R group with the opposite charge, hydrophobic exclusion of nonpolar group, R group, and covalent bonds. Quaternary structure, when two or more polypeptide chains associate to form a functional protein, the individual chains are referred to as subunits of the protein. The primary structure of protein, as is shown in this figure, is its amino acid sequence. Secondary structure results from hydrogen bonds forming between nearby amino acids. This produces a beta-plated sheet and coils called alpha helix. So in secondary structure, we have two arrangements of the primary structure of protein. The tertiary structure is the final three-dimensional shape of the protein. This determines how regions of secondary structure are then further folded in space to form the final shape of the protein. The quaternary structure is found only in proteins with multiple polypeptides. In this case, the final structure of proteins is the arrangement of the multiple polypeptides in space. We have more than one polypeptide. Each of them represent part of the quaternary structure. As biologists discovered the three-dimensional structure of protein, they noticed two classes of similar proteins, motifs and domains. Motifs, which is a common element of secondary structure seen in many polypeptides. A protein sequence, motifs, is an amino acid sequence, patterns found in similar proteins. Changes of a motif change the corresponding biological function, and it's useful in determining the function of a known protein. Domains of protein are functional units within a larger structure. They can be thought of as structure within the tertiary structure of protein. To continue the metaphor, amino acids are letters in the protein language. Motifs are words or phrases, and domains are paragraphs or even chapters in a book. Most proteins are made up of multiple domains that perform different parts of the protein's function. In this figure, as you see here, the elements of secondary structures can combine or fold to form motifs. These motifs are found in different proteins and can be used to predict function. Proteins also are made of larger domains, which are functionally distinct parts of protein, and the arrangement of these domains in space is the tertiary structure of protein. Biologists thought that newly made proteins fold spontaneously. Protein's primary structure, or the amino acid chain, folds its way toward its final form, nonpolar. Sticky proteins are exposed during intermediate stages. If these intermediate forms are placed in a test tube in an environment identical to that inside a cell, they stick to other unwanted protein partners, forming a gluey mess. So how do cells avoid having their protein clump into a mess? So normal cells contain chaperones proteins, which help protein fold correctly. Some disease result from improper folding, such as cystic fibrosis, is a hereditary disorder caused by mutation in the gene of proteins. So in cystic fibrosis, which is a hereditary disorder caused by a mutation in the gene for a protein that moves ions across cell membrane. As a result, people with cystic fibrosis have thicker than normal mucus, resulting in serious lung and organ problems. The most common variant is the deletion of single amino acids, which affect the protein's ability to fold properly. This results in a large number of misfolded proteins. The barrel-shaped structure represents a chaperone. An incorrectly folded protein enters one chamber of the barrel end, and a cup seal the chamber. Energy from the hydrolysis of ATP fuels structural alterations to the chamber, and this will change the unfolded protein from hydrophobic to hydrophilic. This change allows the protein to refold after a short time. So then the protein is rejected, either folded or unfolded, and the cycle can repeat itself. If a protein's environment is altered, the protein may change its shape or even unfold completely. This process is called denaturation. Denatured proteins are usually biologically inactive. This action is particularly significant in the case of enzymes because every chemical reaction in a living organism is catalyzed by a specific enzyme. It is vital that a cell's enzyme work properly. Protein can be denatured when the pH, temperature, or ionic concentration of the surrounding solution changes. Note that these environmental changes affect the interactions of protein surfaces, leading to the loss of structure.